Welcome, everyone. It is my pleasure to introduce Josh Saines as this year's Scolnick Prize winner. The Scolnick Prize was endowed by a generous gift from Merck Pharmaceuticals in honor of Ed Scolnick, who was president of Merck Research Labs for 17 years. I'd like to acknowledge the members of the selection committee, some of whom are here on the Zoom seminar, and that includes Bob Horvitz, Ann Graybeal, Nancy Kamwisher, Story Landis, Joe Coyle, Catherine Dulac, and myself. Now, Josh asked me to skip his biography because he's planning to describe some of his history in his own talk. But I just have an idea he's not going to be talking about his own many awards and honors. So I'll just mention a couple here, that he's a member of the National Academy of Science, the American Academy of Arts and Sciences. He's received the Alden Spencer Award, the Gruber Neuroscience Prize, and the Cowan Award for his work in developmental neuroscience, among others. Now, Josh is one of a small group of some of the most distinguished figures in neuroscience who have discovered most of what we know about synapse development and function. Beginning in the early 1980s, he pioneered the use of molecular and cellular biology to understand the mechanisms that govern synapse development at the neuromuscular junction, including the analysis of intercellular signals and intercellular responses that coordinate the formation and maturation of the synapse. He worked out the mechanisms by which the presynaptic cell helps orchestrate postsynaptic differentiation and how the postsynaptic cell promotes presynaptic differentiation. He studied how a given neuron forms a synapse with the correct target among many possible choices in development, including the key role of the N-coherins and synaptic adhesion molecules. Altogether, he and his colleagues, and I just lost my place here. Altogether, he and his colleagues provided much of the molecular support for the chemoaffinity hypothesis pioneered by Roger Sperry for how developing neurons find their appropriate partners during development. During all this work, Josh also became a major technical innovator in neuroscience, introducing many of the molecular and cellular methods used by neuroscientists around the world, including many of the people on the Zoom call, including the recombinant retroviruses for tracing cell lineage and fluorescent transgenic mice for visualizing neural connections, including the now famous rainbow mouse. Finally, in the most recent phase of his career, Josh has become one of the most foremost anatomists of the retina, using single-cell transcriptomics to identify many previously unknown cell types, and in a series of really elegant studies has started to bridge the gaps between gene expression, anatomy, neuronal function, and even behavior. So we very much look forward to hearing Josh tell us about his work directly. Josh.
Well, thank you very much. I'm really honored, as you might imagine, by the receipt of this award, and pleased to have the opportunity to tell you about some of our research. It's especially meaningful in view of Ed Scolnick's many contributions and those of the eminences who preceded me. Before I start, I also want to note how much I admire the neuroscience community at MIT generally and at the McGovern Institute in particular. As we worked at Harvard to build CBS and HBI, you guys have been an inspiration, and although I shouldn't say it, a source of occasional envy. Now, Bob suggested that I include some background before telling you about recent projects. That's why I asked him to minimize that part. And I decided to put together a microscientific autobiography as prelude to what you might rather hear. And as I did that, I realized that my scientific journey, I hadn't quite appreciated this before, had a strong ascending phylogenetic component. My first laboratory experience, in fact, was in late high school and early college in the laboratory of a wonderful man named Robert Guthrie in Buffalo, where I grew up. And he had developed bacterial assays for detecting inborn errors in metabolism, in particular phenylketonuria. He was ahead of his time in promoting science education for students, and regularly invited students into his laboratory and gave us the quite unusual opportunity to actually be useful screening Bacillus subtilis mutants that could be used in new tests. Then towards the end of college, I had the even more extraordinary opportunity to work on protein kinases in the laboratory of Paul Greengard, who was not only one of the greatest molecular neurobiologists of the last century, but also a brilliant and loving man who remained a friend and mentor to me until his death just under 2 years ago. Now, I went to Paul Greengard's laboratory because my interest in neurobiology began with a desire to learn something about the biological basis of mental illness, which was really also what motivated Paul. When I got to graduate school, though, it became apparent that available methods really weren't up to the task. And so I turned to developmental neurobiology with a sort of vague idea, which now turns out to be likely true, that many psychiatric diseases may be neurodevelopmental diseases, particularly diseases in which something goes wrong with synaptic development. So for my PhD thesis, I climbed up to Manduca sexta, working on sensory neurons and their development as the Manduca caterpillar turns into the beautiful Manduca moth. And my advisor there was John Hildebrand, who I always thank for being especially kind and understanding of me at a time when I was far more obnoxious and high-strung than I am today. Now, during thesis, I took advantage of the opportunity to get closer to synaptic development by arranging a postdoctoral fellowship that began with work on the neuromuscular junction of the frog Rhinopippians with Jack McMahon, and continued with molecular inquiries into the neuromuscular junctions development with Zach Hall at UCSF, now turning to rodents, rats and mice. And I've continued to use mice as our main model animal ever since, although recently, I must say, we've begun to look at non-human and human primates, but I hasten to add only postmortem and only their eyes. Now, starting with postdoctoral work and continuing as a faculty member at Washington University and more recently at Harvard, I focused really in large part on synaptic development with just small forays into other developmental areas such as neuronal lineage and migration. And for many years, decades really at WashU, we stuck with the neuromuscular junction, but eventually moved up the neuraxis to the retina, which has become our exclusive model over the past decade. Now, the reason we use the neuromuscular junction, the synapse made by motor neurons on skeletal muscle fibers, is that despite the fact that it's a peripheral synapse and doesn't even have a neuron as its postsynaptic component, as a muscle fiber, it has advantages that just were not shared with central synapses that we and everybody else would have far rather studied. Those advantages include accessibility, a lot of knowledge about its structure and function, maybe preeminently its large size, which made it possible to assay a lot of developmental manipulations by light rather than electron microscopy. And I think you can see that here on the right with a micrograph of a neuromuscular junction in a mouse and 3 hippocampal synapses in a mouse, all at the same magnification. And we use this system then to look into initially a phenomenon that had been described a century earlier, actually. And that is that when one cuts the motor nerve supplying a muscle, the axons regenerate and form new synapses, with more than 95% of them forming at original synaptic sites, even though those sites occupy only 1/10 of a percent, between 1/10 of a percent and 1% of each muscle fiber's surface. So this is a remarkable example of subcellular specificity, and as such, was something I really wanted to look into, given this interest in how synapses wire up the nervous system, and ultimately how they may miswire in ways that could explain some behavioral or even neurological diseases. So we looked into this. Again, this began as a postdoctoral fellow, and found that some of the critical determinants that axons recognize are not in the muscle or on its plasma membrane, but rather in the synaptic cleft that intervenes between the nerve terminal and the postsynaptic muscle fiber membrane, a basal lamina that's specialized in this particular region. Now, at Washington University, this work gave us a place to look, and we began looking for molecules within the synaptic cleft that could account for this precise reinnervation. And we were then able to look at roles that these molecules and their receptors played in normal synaptic development. And that project was made possible thanks to a long, intense, and very life-altering for me collaboration with John Murley that lasted for more than a dozen years until his untimely and sudden death from a heart attack in 1995. Now, John was a pioneer in applying genetic engineering to neuroscience, generating transgenic and then knockout mice to answer questions about neural development. He taught me all I know about that. And then starting together and continue after his death, we ended up analyzing something like 40 genes using knockout technology, ones that encoded components of the synaptic cleft, which is where we started, some of their pre and postsynaptic receptors, and components of their signal transduction pathway. And putting that all together, we were eventually able to come up with some sort of rudimentary idea of how it is that the initial contact between a motor axon's growth cone and a newly formed myotube turns into this elaborate, highly specialized adult neuromuscular junction. What are some of the iterative intercellular interactions between the nerve and the muscle that pass information back and forth, often using the synaptic cleft as a molecular bulletin board to pattern the formation of the eventually mature neuromuscular junction. Now, as this work came to fruition, not to say it's finished, there's still many unanswered questions, I became interested again in going back to what had fascinated me initially, which is the issue of synaptic specificity, how it is that an axon entering into an area with many potential postsynaptic targets manages to find one particular one on which to form synapses, thereby generating the information processing circuits that we all want to understand. We tried to use the neuromuscular junction for this purpose. Guoping Feng was part of one of those efforts. And the results were interesting, but it pretty soon became apparent that to really look at how neurons wire up in specific ways to form neural circuits, we needed to study more complicated neural circuits. And so we pretty rapidly settled on the retina as the model to study. So the retina, as you know, is outside the skull, but is part of the brain. It is not a piece of film, as people thought once, but rather comprises many cell types that establish complicated and highly stereotyped circuits. And just like the neuromuscular junction, it affords advantages that make it possible to ask questions at a level of detail here that you would like to ask about areas like the cortex or the hippocampus or the cerebellum, but can't really because of technical reasons. It's more accessible. It has clear borders. You know exactly what you're cutting out. It has no long range inputs. It has a very well organized set of laminae and sublaminae in which the neurons are arranged and so forth. And so, as I said, that has become our model system of choice and our exclusive model for the past decade. So let me give you a quick sense of how the retina works. It comprises 3 layers of cells separated by 2 layers of synapses that are called plexiform layers. The outer layer has rod and cone photoreceptors. They form synapses on interneurons that are of 3 classes: horizontal, bipolar, and amacrine cells. I'll say more about them in a minute. And those cells make synapses in the inner synaptic layer on retinal ganglion cells, whose axons run through the optic nerve to other centers of the brain for further processing. Now, the reason this is a great model for studying circuit development and synaptic specificity is that there are actually many types of interneurons and many types of retinal ganglion cells, so that in a mouse, dozens of types of neurites intermingle in what at birth is a very, very small space. So everybody's within filopodial reach of everybody else. And then they sort out to form the complex circuits that enable retinal ganglion cells to no longer be light detectors, but rather feature detectors with each type selectively stimulated by some visual feature like motion in a particular direction, color, contrast, edges, and so forth. And so we set out to ask whether we could find some of the recognition molecules that enable this specific connectivity, and took a transgenic approach, generating or obtaining transgenic lines that would selectively mark one or a few of these retinal cell types, giving us access so that we could look at them, target them for physiological recording, look at their morphology, access them so that we could manipulate their genomes, and sort them, isolate them, and purify them by FACS so that we could ask what sorts of recognition molecules they expressed. And over a period of, I would say, almost a dozen years, we ended up identifying a set of recognition molecules and transcription factors that play some roles in setting up the circuitry that endows three types of retinal ganglion cells called W3B, JRGCs, and OODs with their feature detecting properties. So this was very few out of the many neuronal types. And in none of these cases could I say that we'd come to a satisfying conclusion. So it became apparent maybe 5 or so years ago that doing things in this retail fashion was not going to get the job done in my lifetime or even in the lifetime of my students. And instead, it was going to be necessary to find some way to comprehensively classify and characterize the neurons so that we would know what all the types were and know what all the important molecules developmentally, the recognition molecules and transcription factors were, and find out, among other things, just how many types of retinal cells there are that we have to deal with. So this was a fantasy that was made real by the discovery of methods for high throughput single cell RNA sequencing. And it was such a great idea that I discovered 3 times independently in 3 different labs, all more or less simultaneously. Once by Evan McCosco in Steve Carroll's laboratory at Harvard, once by Alon Klein and Mark Kirshner's laboratory, also at Harvard. Interesting story there. And the 3rd time by a group that eschewed high-profile publications, but instead started a company and marketed what's called the 10X system, which is the one that virtually everybody uses now. And at this point, they could probably buy, sell, press. At any rate, I've put Drop-Seq in color here because we got involved at an early stage in Evan's project because he saw the value of using the retina as a way to perfect the method and validate it based on the features I've mentioned, that we know so much about the cell types and what genes they expressed. And so at this point, I think everybody knows how single cell RNA-seq works, and probably half the people in the audience use it. So I'll just give you sort of a 30-second, 1-slide introduction. In essence, the method involves dissociating a tissue, isolating the cells, or more recently the nuclei, and passing them through a microfluidic device in which a single cell gets encapsulated in a nanoliter-sized oil droplet with a single bead. And those beads then have the cell and the particle in lysis buffer. The cell immediately lyses. Its mRNA is captured by oligonucleotides on the bead, and the emulsion is then broken, and all of the messenger RNAs are turned into cDNAs, amplified by polymerase chain reaction, and sequenced together in a single tube. And that's really the key to this method. Instead of needing to, let's say, sequence 2,000 single cells with 2,000 test tubes and an untold number of graduate students and hours, you can do the whole thing in a single tube in a more or less automated way. And then computationally, there are ways to separate out the transcripts and pool them into those that came from each individual cell. So here is a display of the first experiment that Evan did on Retina. It's the compilation of about a dozen runs totaling almost 45,000 cells. And this is a t-SNE plot that many of you are probably familiar with, in which something like 44,808 dots are arranged. And their arrangements and their colors tell you what the computer thought were cells that were transcriptomically similar enough to be considered candidates to be single cell types or small groups of closely related cell types. And because this was the retina, we could look at genes selectively expressed by each of these clusters and ask whether they made sense as individual cell types. And to make a long story short, they did. One cluster would be rods, one cluster would be cones, several clusters would be amacrine cells, and so forth. So it worked, but it was limited. And you can see that most clearly in that there were only 39 clusters. And we already knew at this point that there were going to be more than that, at least 60 types of retinal cells. And the problem was that even though Evan was profiling an immense number of cells, the vast majority of them, 70 to 80%, were rod photoreceptors. And so there weren't enough cells in this population of the very rare types, especially rare types that were similar to other rare types, to have the computational power to separate them into individual clusters or cell types. And so what we did then was to go on and develop transgenic methods to isolate the three most heterogeneous classes that I mentioned already: the bipolar interneurons, the amacrine interneurons, and the retinal ganglion cells, and isolate enough of them to take the project to a conclusion. And here's what we found over a few years: a total of 15 types of bipolar cells, 46 types of retinal ganglion cells, and an absolutely terrifying 63 types of amacrine cells. So right away, you get some evidence that what I said is true, that the retina really is as complicated as almost any other part of the brain. These are similar numbers to what the Allen Institute finds in motor cortex or visual cortex. And we were able to go into each of these clusters, and just as we did with Evan, find genes selectively expressed by one or another of the clusters that's shown in these dot plots, and then use in situ hybridization and immunohistochemistry to ask whether the molecularly defined clusters had a morphological homogeneity or a morphological identity, and in some cases a physiological identity, since clearly cell types defined by purely molecular criteria would be of very limited interest to most neurobiologists if they didn't correspond to cell types defined by things that most neurobiologists care about. And the answer basically was good. For the retina, at least, in every case we studied, which is not all of them, but many of them, we were able to find a one-to-one match of molecules to morphology to physiology. And we can come back to this in discussion. It's not entirely clear that this will be true of other areas like the cortex, in which so much development is experience dependent, but it is true in the retina. So to summarize that early work then, by a couple of years ago, we had an atlas of the mouse retina comprising around 130 neuronal types and around 10 non-neuronal types, so a total of 140. And it's interesting to note that the number of types identified and characterized about doubled over a period of 10 years. And of course, lots of people were working during that decade, including some beautiful work on high throughput electron microscopy. But I think it's fair to say that the biggest increase came from single cell transcriptomics. And at this point, one would never be shocked to find 1 or 2 or 3 or 4 more cell types, but I believe we have a fairly substantial and reasonably complete cell atlas of the mouse retina. And the fact that we can do that and show this harmonization of molecular, physiological, and functional criteria, I think has helped people in other areas that are less accessible and more difficult to deal with to go on and try to continue this enterprise there. So as this came to completion, we decided that we would try to use the atlas to do some interesting biology. And over the past few years, we've worked in 5 areas that I'll just describe very briefly and then sort of focus in on a few of them. So the first is development, which is, of course, where we came from. A second is selective vulnerability. Can we use the multiplicity of cell types in the retina to look into the very general issue of why, following injury or neurodegenerative disease, some different cell types are able to survive or regenerate better than other types? And ultimately, can we use that information to figure out ways to make vulnerable genes more resilient? We also asked whether we could use the mouse atlas as a starting point to look at human retina with a stop along the way with non-human primates. And the particular question here was to see if we could reveal special features of the fovea, the primate specific, at least amongst mammals, central part of the retina that mediates high acuity vision. And with those atlases in hand, we could look at where genes that have been implicated in diseases of the retina are expressed, what cells in the retina, and we've gone on to now look and develop an atlas of the entire rest of the eye, so we can ask whether genes that predispose to retinal disease are expressed in retinal neurons or perhaps neurons in the anterior segment, the trabecular meshwork, the ciliary body, and so forth. And then finally, we're just beginning to look at the evolutionary question, using the retina as a model to ask how conserved are cell classes, and by that I mean, in this case, photoreceptors, interneurons, output neurons, and how conserved particularly are cell types among vertebrates. A question not unlike that asked about genes, but it can also be asked about cell types. So today, I'm going to focus primarily on the first 2 of these questions, and depending on how fast I talk, say just a little bit about evolution at the end. So for development, there are many questions about diversification, expression of adhesion molecules, transcription factors, effects of experience on the development of the retina at the molecular level. And we've looked at all of these, but I'm going to focus here just on the one on how cell types diversify. And so by background, let me say just a few words about how these cell classes in the retina diversify. And we know quite a bit about that because more than 30 years ago, 3 groups, Scott Fraser's, Christine Holt and Bill Harris's, and Connie Sepko's here at Harvard, showed in different ways and in different species that there are multi-potential progenitors that can divide and give rise successively to the different cell classes: retinal ganglion cells, horizontal cells, rod photoreceptors, amacrine cells, cone photoreceptors, and so forth. And they do that in roughly the birth order that was already known. So the first progeny are cones actually in retinal ganglion cells. The last progeny are rods and Mueller glial cells. We also have some idea of how this progenitor changes. And the most influential model there is Connie Sepko's competence model, in which she posited, and she and many others have now gotten evidence for the idea that these progenitors change their state as they move along and throw off postmitotic cells. And as a consequence of intrinsic changes and extrinsic influences coming perhaps from earlier born cells, they become capable now, or maybe even committed to, generating different cell classes. And then I say the third big idea is one that maybe Bill Harris is most responsible for. And that is the idea that the choices are maybe not completely deterministic, but rather stochastic. That what's happening as progenitors mature is that they change the frequencies with which they're likely to throw off particular progeny. So at first, they're most likely to give rise to a retinal ganglion cell, much less likely to give rise to other types, at the end the other way around. So I mention all this to draw a contrast with what we know about the generation of cell types, the 46 types of retinal ganglion cells, the 63 types of amacrine cells, and so forth. And there we know nothing about the progenitor, nothing about competence, nothing about stochasticity, partly because people didn't really know what the types were when they were doing these initial classic experiments, partly because to the extent they did know the types, they could define them only physiologically, which wasn't much use for an inquiry like this. And so we decided to see if we could use our atlas as a basis for looking at the diversification of one class, the retinal ganglion cells. And the way we did that was to first ask about two extreme models. So we're now looking post-mitotically, and we posited two extreme models about how these ganglion cells could diversify. We here being Karthik Shekhar, who led the computational effort, and Irene Whitney, who led the experimental effort. So one extreme would be that there are actually 46 types of progenitors, each of which is committed to give rise to a particular type of ganglion cell, and then go on to give rise to a host of other cell classes and types, either in a deterministic way or not. And the other extreme would be that all ganglion cells initially post-mitotically are identical, and then they diversify in some stepwise fashion, eventually forming all the 46 types. And so the way we tried to look at those extremes and points in between was to look first at the adult atlas, and then go earlier, and ask how many clusters we saw by similar and other computational methods at each time, starting from embryonic day 13, which is the earliest time at which a large number of retinal ganglion cells are becoming post-mitotic, all the way through to adulthood. And the upshot was that the number of clusters increased over time, increased several fold, and the clusters became not only more numerous, but became more clustery. That is, they become transcriptomically more separated from each other, a point I'll come back to in a minute. And so these data, along with a number of other considerations and controls that I won't go into, let us tentatively exclude the model of there being 46 types of progenitors, each initially committed to a particular type. So it was either stepwise or somewhere in between. And we next asked whether that stepwise diversification was specific and determinate. That is each individual post-mitotic cell committed to give rise to a small set of adult types, or is it non-specific? Are the individual cells initially capable of giving rise to a number of cells with overlaps amongst the early cells in what adult progeny they can eventually give rise to? And one simple way to distinguish those alternatives is to transcriptomically match the early clusters to the late clusters. And in a specific and deterministic scheme, you would expect that each early cluster would share most of its transcriptomic similarity with a specific set of adult types, whereas there would be no such relationship if there was complete non-specificity. And so you'd look at that, and here are just two examples, and the upshot is no, it's not completely random, but it's certainly not completely deterministic. The initial transcriptomic types here, clusters, I should say, here looking at embryonic day 13 and 14, seem to share transcriptomic similarity with many different adult types. So again, we can tentatively rule out a very specific model and say instead there's some sort of non-specificity. And then next and last for this part of the talk, we wanted to ask what this sort of non-specificity was. And what I mean by that is the analysis I just described is at the level of clusters. So we see a level of indeterminacy at the level of clusters, but it's possible that the determinacy is much greater at the level of individual cells. That it could be that the early clusters are made up of partially overlapping sets of individual cells, each of which is determined to give rise to one or a very few eventual adult types. Alternatively, it could be that even the individual cells within the clusters are indeterminate and multipotential. Now, the only way to really answer that question in a satisfying way is to find a way to visualize a postmitotic cell, follow it through to adulthood, figure out what it is, and then run time back in a time machine and do it over again several times and see whether a cell is truly multipotential. Now, at least in my lab, that isn't yet possible. And so we took an indirect computational approach using a method called optimal transport, which was actually invented more than a century ago for completely different purposes, but was then repurposed by Eric Lander and his colleagues at the Broad Institute to be used with transcriptomic data. And the basic idea is to look at a cell-by-cell basis at how the transcriptomic mass of an individual cell is apportioned to other cells at later stages. So it's a sort of computational lineage diagram. It is inferential, but it's the best we can do. And using that method, what Karthik found was that indeed the newborn retinal ganglion cells are very multipotential. Each one gives transcriptomic mass to something like 10 or 12 adult types. And then that potential decreases over time until it becomes 1 in adulthood, because at that point, of course, every cell has become what it's going to be. And the diagrams here on the right are meant to show you that it's many, many cells that are multipotential, most of them not just a single small subset of cells. So at least inferentially, we can rule out this idea of there being cohorts of determined cells, but rather conclude that cells are truly multipotential at an early stage, and their potential then decreases in a stepwise fashion until they eventually, each individual cell matures into one of the 46 types. Now, with this model in mind, we can begin to look at whether the groups of individual cells end up maturing into adult types that are related to each other. And one of many ways of visualizing that is in what's called a Sankey plot, in which one plots the, let's say, the paths of maximal transcriptomic mass from groups of cells now calculated by optimal transport to groups of cells at each subsequent stage. And to make a long story short, the answer is sort of. For some subclasses of retinal ganglion cells, ones that express TBR2 or NeuroD2 in adulthood, there seems to be a very restricted group of early progenitors that mature into those types via stepwise diversification and fade uncoupling. For other types, there's a less clear pathway. And so the idea now is that one can use Sankey plots and go back to these early stages and look for transcription factors that might be expressed at those early stages that are potential fate determinants of groups of adult types. And of course, with those transcription factors in hand, it ought to be possible to use mouse genetics to test those ideas and find out whether there is something about these early multipotential, but maybe partially committed progenitors, and ask what happens if you manipulate them or knock them out. So let me move on to the second topic now of selective vulnerability. And the model here is one of optic nerve crush. And there we've done a series of experiments in close collaboration with Zhiguang He, who is a real expert in this area. His lab is at Children's Hospital. And the paradigm is that if you crush the optic nerve in a mouse and sever the axons, 80% of the ganglion cells die within 2 weeks, and there's almost no regeneration of axons. Some limited regeneration can be provoked with a number of interventions, several of which have been pioneered by Cajal. And so what we started by asking is whether this survival is a stochastic process. That is it that some cells live and some die, maybe because they were active when the nerve was crushed, or because they were near a blood vessel, or because they had a little more trophic factor, or could there be a cell type basis for selective vulnerability? At the limit, could it be that 80% of the types completely die and 20% of the types completely survive? So in the first step, we used some of the transgenic lines that I mentioned, and indeed found dramatic differences amongst groups of types. So a subclass called alpha ganglion cells survive very well. One of the intrinsically photosensitive ganglion cell types survive very well, and other types survive very poorly. TUJ1 is a marker for all the types, just confirming that only 20% of the types were left at this period 2 weeks after nerve crush. So with the cell atlas in hand, we decided to ask this question of all the cell types, and more importantly, find out what those cells were expressing and whether there were differences between the vulnerable and the resilient types. And so the way we did that was calculate the frequency of all 46 types based on our single cell data. And we cross-checked that against a lot of histological analysis. So we had some confidence in the frequencies determined by transcriptomics. And then we cut the optic nerve, did the whole thing again 2 weeks later, and then divided one by the other, looking at the fraction of each of the 46 cell types that survived over 2 weeks. And there were two main findings. One is there are indeed dramatic differences. Some resilient types survive to the tune of almost 90%. Some vulnerable types die to the tune of almost 95%. And second, it's not a binary division. There's rather a continuum between the most resilient and the most vulnerable types, something we didn't really know from the original study because we were able to look at so few types. So we could then look into the RNA-seq data and look for genes that would be selectively expressed by resilient or susceptible types, either before injury or at each of 6 time points that we looked at after injury. And the news was good and bad. The good news is that there were some types that were expressed in some resilient or some susceptible types, but not the other way around. The bad news was that we found no genes at all, sadly, whose expression was confined completely to all resilient types and no susceptible types or the other way around, nor any genes whose level of expression correlated with resilience or vulnerability. Nonetheless, we were able to pick some promising candidate resilience genes and knock them out, or some promising candidate vulnerability genes and overexpress them in resilient cells using AAVs for overexpression and an AAV-based CRISPR method for knockout. And indeed, in some cases, both improved ganglion cell survival. So it wasn't a huge effect. None of these are running to the clinic, but it was some validation that this could be a good method for identifying targets that might ultimately be of some therapeutic value. So I'll tell you now what we're doing as a next step. And this, again, is work by Nick Tran and Jacoby and Wenjun Yan. I forgot to mention that Karthik Shekhar was involved in the first step. And the idea here is to now go the next step and ask how these therapeutic interventions improve ganglion cell survival and regeneration, with a hope of finding out something about the mechanism underlying these effects. And so what we've ended up using are 3 interventions that Zhigong and his colleagues have pioneered, which are very robust and been repeated multiple times. One is enhancing mTOR signaling by deleting the mTOR endogenous inhibitor P10. One is enhancing JAK-STAT signaling by deleting conditionally its endogenous inhibitor, SOX3, and a third is overexpressing CNTF. And you can see here the enhanced regeneration in these interventions, and here from one of Zhigong's papers, the enhanced axonal regeneration, in this case in the double knockout conditional of P10 and SOX3. So the idea then has been to do these interventions and then isolate retinal ganglion cells for single cell transcriptomics and see if we can find what genes are expressed in the cases where there's been intervention and not otherwise. Now, we've recently augmented this inquiry with a beautiful method that Anne Jacobi came up with, in which she's able to actually selectively label in a retina those cells that regenerate from those cells that survive but don't regenerate by, in a really arduous surgical manipulation, injecting a fluorescent tracer in the optic nerve so it'll be picked up by regenerating axons, but not by those axons whose stumps have survived but haven't regenerated. And you can get a sense of that down here. Here's the fluorescent tracer. And in this whole mount, these little red dots are the retrogradely labeled regenerating ganglion cells, many controls that I'm not going into. And so what Anne can now do is use two labels to isolate those retinal ganglion cells that have regenerated from those that survived, but most likely did not regenerate. And the types there are actually quite interesting. I'll say just a word about this. In this double conditional knockout with CNTF, it looks like actually almost all of the types that survive regenerate. And why that's interesting is it may be that in this case, regeneration is not cell type specific, but there's something magic happening to some of the cells of many types that changes their state and allows them to regenerate. Now, whether their cell state is the cause or the consequence of regeneration, of course, remains to be worked out. But at any rate, looking into the genes, the idea here is to combine all the interventions, all the types, the crush with no intervention, and then recluster the cells in a number of ways. And Anne, Nick, and Wenjun have tested 4 different methods. In one, they cluster cells by the type of intervention. In other, by their transcriptomic similarity overall, whether it be cell types or states. Another in which they emphasize states without cell type labels, and another in which they're clustered based on what are called regulons, that is, co-expression of transcription factors and expression of genes that are predicted to be the targets of those transcription factors. And this analysis is still underway, but remarkably, the preliminary result is that all 4 methods have generated a set of similar modules, something that I never would have expected, yet seems fairly likely to be true. And I'll show you just one example here by the regulon method. And so here are the cells sort of sloppily reclustered by that method. And here are 3 modules, that is clusters along the bottom, sets of genes along the right. And you can see that these 2 clusters selectively express this set of genes here and here. And you can see the cells that contribute to those modules, numbers 1, 2, and 3, are largely, albeit not entirely, non-overlapping groups. And so here are the 3 modules. Module 1 is enriched in genes that evoke cell death and apoptosis, like DDIT3. And the cells that contribute to that module are enriched in the vulnerable ganglion cell types and types that appear to be on their way out. Module 2 down here is enriched in genes that have been implicated in neural development, for example, ALCAM and BDNF. And the cells in this module, whoops, over here, are enriched in, looking at Anne's results from the retrograde labeling, cells that appear to be surviving but not regenerating. And then module 3, in which cells, the cells contributing to that module are enriched in the regenerating group, are also enriched from a transcriptomic level in genes that have been implicated in axonal growth and regeneration. So the idea now is to look particularly into this regeneration module and ask whether manipulating any genes in that module can actually promote axonal regeneration. And so here are 3 genes that Anne and Nick have tested so far. You can see their expression in regenerators compared to survivors from Anne's retrograde labeling data confirms their identity as being selectively expressed in those ganglion cells that regenerate over those that survive but don't regenerate. And indeed, the initial data suggests that all 3 of them, when overexpressed using an AAV, can promote some level of axonal regeneration. So I've left myself 5 minutes, and that's good because I now get to say a few words about evolution. And as I said, the idea here is to do or initiate a project to do for cell types more or less what people have done in so much detail and so beautifully for the conservation and evolution of genes and gene families. And we think the retina, again, is a good place to do this because its basic plan is conserved essentially amongst all vertebrates. They pretty much all have the same 6 classes of photoreceptors, 3 types of interneurons, ganglion cells, 1 type of intrinsic glial cell, the Mueller glia. And so what I've told you about so far are mouse transcriptomics, but we've expanded that now to look at the retinas of a large number of other species, a number that I hope continues to grow. We have incomplete cell atlases for them, but enough to begin to look at evolutionary conservation of at least some of these cell classes. And so if we look first at the classes in a 4-way comparison that Wenjun and Yan has done, looking at chick, human, macaque, and mouse, what we can see is that what was known to be the morphological and physiological conservation of classes is perfectly maintained at the molecular level. So for example, a bipolar cell of a chick is transcriptomically more similar to the bipolar cell of a mouse, macaque, or human than it is to any other chick retinal class. So that's good, that's gratifying. And now really the big question, where we've only begun, is to ask what happens to conservation at the cell type level. And here's just an initial result. So reclustering in these 4 species, we can ask the degree to which types are conserved among species. And one very simple initial metric is to ask in the reclustering how many species contribute to each of these clusters. And I think if you look just at the colors here, you can get a sense that there's more mixing for the amacrine cells than for the retinal ganglion cells. And that's borne out arithmetically. The number of species that contributes to each cluster is greater for all of the 4 other neuronal types than it is for ganglion cells. There are many confounds here about type number and so forth, but I think the overall conclusion is pretty robust. And so it sort of leads to a model in which the outer retina is a conserved set of processors that's been evolutionarily conserved, and where evolution may be acting at the most is at the level of the retinal ganglion cells, where this processed information is now transferred to the rest of the brain. And it may be in the variation among retinal ganglion cell types that we will see the most evolutionary tinkering to, let's say, match the capabilities of each animal's vision to the ecological niche that it occupies. So with that, I'm at an end. I want to thank the people who have done the work. I've put in green here those who contributed to the unpublished work that I described, but it's been a big group. Included in this list are people from collaborators labs, of which there are many. This is the most collaborative project I've ever been involved in. And I just want to say what a pleasure it's been to collaborate with these other terrific scientists and the students in their laboratory. And I always end with a picture of Cajal, but it's Particularly, I would say, suitable and appropriate here, because he, in around 1880, started on the project of cell type classification. And it's sort of fun to think that we are using different tools to do his bidding even now. So thank you very much. So I will stop sharing, and there we go.
Well, let me first of all let me start a round of applause. Can we do this? I don't see an applause button on my Zoom but I will start applauding you with my real hands for that absolutely wonderful, really clear talk, Josh. That was fabulous. And you know, the thing about Cajal was he really he was a superb anatomist, but he was always thinking about function. Like, what does this all mean? And you know, and you are in that same class of anatomists. You want to understand, what does it all mean in terms of circuits and development and now even evolution. And so you're definitely following in his footsteps. Yeah. So we're going to have some questions and answers. Questions, and Josh will give answers. And so the first question is from Fan Wang. I think we can unmute you. Can we try doing that?
Can you hear me?
I can.
Yeah.
I just hi Josh, beautiful talk. Just wanted to ask whether or not activity, do you see activity playing any role at all in cell type specification?
So I didn't talk about it, but we block visual activity postnatally in a couple different ways, 2 different mutants, and just by dark rearing from birth. And of course, a lot of diversification is done by birth, but we see no effect on the remaining diversification. And maybe more surprising is that the effects on gene expression are shockingly modest. And we know that activity plays a huge role in the central wiring of retinal ganglion cell axons. And so we just need to look into what those genes are that are affected by activity, and we haven't done that yet.
Great, thank you.
Just maybe I can ask you a question about the regeneration, the regeneration work in the axons. Are you seeing this as a way of understanding more about the programming of the cell identity, or are you actually thinking about this therapeutically? That there's you might be a way of promoting axonal regeneration.
I guess, I mean, first, as a basic science question, the mechanism is fascinating, and it's a general question for all neurodegenerative diseases, right? I mean, you know, why do you have, let's say, an SOD1 mutation in all your neurons in ALS, but motor neurons selectively die? But yes, I think there's some potential therapeutically. We looked, of course, to see whether after injury, the cell type identity is compromised. Do they lose their type specific features? And by and large, no, they don't.
We have a question from Ayano Matsushima. Maybe I can just read the question. The question is the resilience susceptibility determined by the genetic profile predetermined at the stage of ancestry progenitor, or expressed just before the injury, or induced in response to the injury? Okay,
so, you know, at least tentatively, we're assuming there's only one type of progenitor. So when the differences appear as stepwise, when in that stepwise progression, the vulnerability is instantiated, we have no idea. Certainly in the very small number of genes we've tested, some are included that are expressed in, let's say, resilient types prior to injury. Others are expressed only after injury. So I think it'll be a mixed bag. But of course, even those that are expressed in particular cell types only after injury are expressed in those particular cell types because of some sort of pre-programming that had to be there before injury.
So thank you. We have a question from Alan Jasanoff. Why don't we just unmute Alan? You can ask this.
Hello.
Hi.
Hi, Josh. Beautiful talk. I wanted to ask you about the relationship between the markers of the cell classes that you've identified and their function anatomy, as you know from other studies, including your own. When you look at the lists of genes that are best, that are most predictive of the different classes that you've found, do they make sense? Do you see channels, adhesion proteins, scaffolding proteins, and so on? Is there any logic that you can decipher maybe post facto from what you found?
I hate to say this, but so far, no. It looks like the genes that are selectively expressed in one type rather than another are everything from, metabolic genes to channels to recognition molecules to pumps. So amongst them, there clearly have to be genes that are important for development and function, but they're not super enriched.
That's neat. Are they definitely different cell types then, as a neurophysiologist would define them?
Well, what we've done is take genes that are selectively expressed, and we just take the ones that are most selectively expressed, no matter what they are, and use in situ or immunohistochemistry to see whether the cells that express those are a specific type by, let's say, morphological criteria. And in every case we've done, yes, they are.
Yeah, I'd say. All right. Well, cool. Certainly food for thought. Thanks.
Kind of a disappointment, frankly. I figured you'd look at these gene lists and the secret of the cell would be revealed, but sadly.
That's what we all want. But I guess the lesson of biology is often that, it's all in statistics and, the deeper logic, the deeper meaning isn't there.
As Mick Jagger said, you don't always get what you want.
Thanks again.
We have a question from Helen Shu, who's actually a postdoc in my lab. Can you comment on how mouse retina seems capable of doing higher functions, for example, direction selectivity, that macaque retina cannot seem to do? Does the mouse retina have more types of interneurons or more complex circuits?
Interesting. Yeah. So the mouse retina seems to have lots more types of retinal ganglion cells than primate retina. And their frequencies are not hugely different from each other, as opposed to primates in which, 80+% of the ganglion cells are midget cells. And so the tentative idea is that for lower vertebrates that send most of their retinal output to the superior colliculus, they do a ton of pre-processing. Whereas when you get a cortex, you sacrifice a few milliseconds in hardwired speed and do more processing in the cortex that gives you more flexibility.
Okay. We have a question from Minqing Zhang, who someone else I collaborate with. Wonderful talk. Do cells with multi-potential differentiation have a spatial distribution feature at a certain age?
So I don't know that, but the retina is very weird in the sense that every little piece of the retina seems to have a complete complement of all 130 cell types, give or take. So that, and of course it makes sense in that it's not like you only see things going up out of the left side of your eye and things going right out of your right visual field. So my guess would be that neither do the progenitors have a major bias to one or another part of the retina.
Okay, thank you. So this will be the last questions from Jill Crittenden, who I work with every day. Is retinal degeneration disorder in retinal degeneration disorders, is there a preferential loss of genes that define the RGC types?
Yeah, so we've only really looked at nerve crush, which is not a big public health problem. We would love to do this in mouse models of glaucoma and actually even in human glaucoma and ask. But in optic nerve crush, there are changes in gene expression, but they do not lose their type identity. They get sick and die, and when they die, they're the type that they are.
Okay. Well, let's give another, Guoping wants to know if he can ask a question live. Guoping, we will allow you this one last live question. live from Newton, Massachusetts. Guoping.
Thank you. Josh, wonderful talk. I was wondering whether across the retina from peripheral to center, the same type of cell type, do you see any differences or topographic differences or gradient or anything gene expression?
So there are some differences in frequency of types. I mean, in primates, of course, the density of cells is much greater in the central retina, and the fovea is very different from the peripheral retina. In mouse, there's a very, very, very modest gradient in density. And for each individual cell type, there may be some gradient in density, some central peripheral, some dorsal ventral. We've seen this with a number of types. Rachel Wong has seen it with a number of types. But they're not huge, really. Nothing that, let's say, challenges the general conclusion that every part of the retina has every cell type.
Thank you.
I'm going to make one more exception because we have a question from our very good colleague, Helen Barbas from across the river. Helen, should we unmute you? No? Well, I'm not quite sure that worked, but she's asking, in the cortex, some neurons in specific areas share features that appear to render them vulnerable to disease. Can you identify clusters of features in vulnerable neurons?
Well, I was showing you our best bet there. And yes, of course, we are hoping that some of what we see would be transferable to neurodegenerative diseases. And of course, people are now doing this a lot in neurodegenerative diseases, Alzheimer's probably more than others. I'd say the last part I talked about, where we're trying to look at gene expression modules after interventions, is partly a response to the fact that although we found interesting genes, we didn't find, perfect markers of resilience and vulnerability. We're hoping maybe that some pathway analysis will be more productive.
Okay, well, Josh, thank you so much. Wonderful talk, wonderful answers to questions. And again, we don't seem to have an applause button here on our Zoom, but just know that we are all applauding you right now.
Thanks so much. Bye-bye, everybody.
Bye-bye. Congratulations again.